Very good afternoon, you all. My topic for presentation is mutations at molecular level. So what are the mutations? Mutation is a process that produces a gene or chromosome. We can differ from its wild type. So these mutations results due to the changes in the gene sequence or chromosome itself. So these mutations are important to know the diversity, what are the changes occur in particular gene or genome of that organisms. So in this mutation we can broadly categorize in two types. So these mutations where alleles of genes can change, and in case of chromosome mutation where the segment of chromosome, or in case whole chromosome or entire set of that chromosome may be affected here. So these mutations are arise due to during replication, recombination or permanent change in the nucleotide sequence of the DNA molecule. So this can be damaged DNA mutated either by substitution, deletion or insertion of particular base pair in gene or genome. So these are the most important where these mutations may form or they lead to cell death, or sometimes there may be a tumor formation. So this is what a lethal potential of DNA mutation. So these cells have evolved mechanism for repairing of these damaged DNA. So in this mutation, these are the change sequence of base in DNA or RNA. These are the important part for the essential for evolution. So in this case, this is what ultimate source of all new genetic material. We can say here the new alleles in a species. So there are different types of mutation. These are based on two different categories. It's a germline mutation and somatic mutation. So germline mutations occur in gamete. These mutations are especially significant because these mutations can be transmitted to their offspring, and every cell in the offspring will have the mutation. In somatic mutations occur in the cell of the body. These mutations have little effect on the organism because they are confined only in a one cell, and it also daughter cells. So these mutations cannot be passed on to their offspring. So these mutations differ in the way of genetic material is changed, and that change the structure of that chromosome of that particular nucleotide. So these are the categories. Body of that organism in case of somatic is localized in mosaic pattern of the cell. These mutation cannot pass down. In case of germline, these are producing gametes. Only mutation is typically in case of germ cell or gamete producing cell. These mutation pass to their offspring, or half of the gene may pass to their progeny. So in case of point mutation, that is the change in a single nucleotide in a DNA. So this change we can see here. This change is very important to know where is the mutation arises. So this type of mutation is usually less serious than chromosomal alteration. In the example of point mutation, changes codon UUU to the codon UCU. So here C nucleotide inserted in place of uracil. So here cytosine is inserted in place of uracil. So that is nothing but point mutation. Only particular gene or coding frame is changed due to insertion of nucleotide. So these mutations can be silent, missense, nonsense, and effect of these mutation depend on how they change a particular genetic code on mRNA during production or synthesis of protein. Base substitution is one of the best term in this is a type of point mutation, because here this mutation only changing one amino acid into another amino acid. So this may also altering beneficial to harmful part of the cell. So these point mutations are the most common type of mutation, are two ways. In case of base substitution, a particular base is going to substitute by another base of another nucleotide. So in these two different types, transition and transversion. Transition type of mutation occurs when purine is substituted with another purine and when pyrimidine is substituted with another pyrimidine. In case of one adenine, adenine is going to replaces to another guanine and cytosine is replaces to thymidine. Transversion, purine is substituted for pyrimidine and pyrimidine replaces a purine. So you can see here transition, T replaces to C, A replaces to G. Means what here? Pyrimidine replaces or substituted with another pyrimidine and purine is substituted with another purine. In transversion, pyrimidine replaces a purine and purine replaces to a pyrimidine. So we can see thymine replaces to adenine or it may guanine also, or adenine replaces to thymine or cytosine also. So we can see here vertical as well as horizontal and crossword the formation of transition as well as transversion. You can see the structure. Adenine replaces to thymine, guanine replaces to cytosine by cross way, or in vertically adenine replaces to guanine and cytosine replaces to thymine in case of transition. Horizontally we can see here adenine replaces to cytosine and guanine replaces to thymine. In the base substitution, this is the gene level type of mutation that involve the swapping of one nucleotide for another during replication of DNA. And these base substitution mutations, only a single nucleotide within the gene sequence is changed. So one codon is affected here, only one codon. So these base substitutions alter only single codon in a gene, and that can have a significant impact on the protein production. So of course, structure of protein will be changed. That results in the part of production of abnormal protein. So in case of point of substitution mutation, codon change, that is because of base substitution, and these lead to different categories. In the first category is what? That is a subcategory. This consists of missense mutation in which altered codon leads to insertion of incorrect amino acid into protein molecule during translation. So here wrong or incorrect amino acid is place of correct amino acid. So due to insertion of wrong or place of incorrect amino acid causes abnormal protein molecule. So second consists of nonsense mutations in which altered codon prematurely terminates the synthesis of protein molecule. So before translation or complete synthesis of protein, codon stop the protein synthesis because that codon is premature. Means what? There is a possibility of insertion of nonsense codon, and that stops the production or synthesis of protein. It is a termination of protein before the complete synthesis of protein. Third part consists of silent mutation in which altered codon codes for the same amino acid as the unaltered codon. So there is a no effect on the protein synthesis because there is an ambiguity of the codon, because here one amino acid is encoded by more than one or two codons. So there is a possibility of what? Altered codon encoded to the same amino acid. So there is a no effect. The effect is totally quenched by the altered codon. So this is what? Silent mutation. So what are the result of point mutation? Silent. This is just because of due to redundancy of the genetic code, and these most mutations are silent. They do not code for different amino acid. They will code for the same amino acid. Missense mutations produces change in amino acid in protein but does not change the function of the protein. And in nonsense produces a stop codon in the midst of the mRNA transcript, and that can produce a non-functional protein. There is a quite possibility to change the function of that protein or may not be function of protein in case of point mutations. So here nonsense mutation is dangerous because premature synthesis of protein is here. These are the sample outcome of DNA code. We can see here the sense strand, template strand and mRNA transcript, and we can see here the translated product of the protein. So we can see methionine, proline, threonine, arginine are the amino acids. Latter stop codon is here. So in case of silent mutation, there is a transition. CG is inserted in place of TA, and there is a no change of the amino acid sequence in a protein. So effect of mutation is quenched. In missense, the part of what base pair is changed means what here? CG is changed by AT. So here amino acid is changed from arginine to histidine. So that is what missense mutation. This kind of mutation produces effective change. Means what? Amino acid sequence in a protein product may change or may not be changed. So this can lead to the change the function of that protein. Nonsense mutation, there is a premature synthesis of protein before the complete synthesis or encoding of amino acids. There is an insertion of what? Stop codon. This is also because of what? Transition type of mutation. So we can see here the complete normal, silent, nonsense, missense kind of mutation. Another mutation is what? Frame shift mutation. This is the fourth type of point mutation. This type of mutation arises from insertion or deletion of particular or nucleotide base pair within the coding region of the gene. We can see here insertion of particular nucleotide or base in a gene or coding region that leads to the change the reading frame of the codon. It's a transcript mRNA. So these frame shift mutations usually are very deleterious and yield mutant phenotype, and these resulting from synthesis of non-functional protein. The reading frame shift often eventually produces a nonsense or stop codon so that peptide product is short as well as different in sequence. You can see here wild type and mutant strain. This can change their coding pattern. So codons may change here in peptide. Instead of tyrosine and tryptophan, valine and leucine is inserted here. So here we can say reading frame is changed because of the insertion. So GC pair is added here. That's why instead of tyrosine, tryptophan, this codon reading frame of this mRNA transcript is formed valine and leucine amino acids. So this reading frame shift produces a different peptide after the addition. This kind of mutation is a type of mutation where involving of deletion or addition of nucleotide, and the number of deleted base pair is not divisible by 3. So divisible by 3 is very important because cell reads a gene groups of 3 bases. Okay. So each group of 3 bases correspond to one of the 20 different amino acids which are used to synthesis of protein. Mutation disturbs the reading frame, then entire DNA sequence following the mutation will be read incorrectly. In a reading frame, one codon may change. Of course, the entire sequence of that region may change here. So this is very important because there is a quite possibility to form premature termination. Instead of encoded protein, being of certain particular size, that is it will end up being much shorter and it won't be able to accomplish the role of that's been set out for it. So we can see here the examples with mutation in the DNA and what type of the change here: silent, missense, nonsense, and frame shift. We can see here the reading frame is totally changed because of frame shift. Another type of mutation is what? Spontaneous. So this kind of mutation is occur under natural in absence of particular knowing mutagenic agent. These mutations frequency occurring is very low or rarely because only one mutation in 10 raised to 9 billion replicated base pair. So there is a very less possibility to cause of mutations. Mistakes occur during replication of DNA just before the cell division or natural error rate of DNA polymerase. So there is a quite possibility during replication DNA polymerase adds wrong nucleotide and forget to repair that part. So here this spontaneous mutation arise variety of sources. We can say replication, lesions, and there is maybe another reason of what? Transposable genetic element. We can see here error in DNA replication, nucleotide, DNA synthesis. These are the important, we can get this information. There is a quite possibility to addition of particular wrong nucleotide. This can be occur due to tautomeric shift because DNA polymerase recognizes its normal nucleotide base because that base is actually form their tautomer or it's a tautomeric form. So tautomeric form are the analogous of the same nucleotide base. But thing is that that nucleotide can pair with the abnormal or unusual Watson-Crick pair. So the ability of wrong tautomer of one of these standard base to mispair and cause a mutation in the course of DNA replication. So this was first noted by Watson and Crick. Induced mutation. This kind of mutation is caused by known mutagenic agent just because of physical as well as chemical mutagens. So we can further subdivide it into different parts. This can be base pair change, base analogous, frame shift mutagens. In physical, UV rays, gamma rays, X-rays, these are the important in the induced mutation. So all these are the reagents responsible to cause of mutations. So ionizing radiations are the alpha, beta, gamma and X-rays, and non-ionizing radiations are the UV rays. These rays are non-ionizing. They produce from mercury vapor lamp or tubes. They also present in solar radiation. So these radiations of ultraviolet sources can penetrate one or two cell layers, and of course the low penetrating capacity, they have ability to only kill microorganism only. So there is a effective treatment against to bacteria as well as viruses. So this UV light causes thymine dimer formation. Means what? There is a formation of what? Intramolecular pairing. So this is nothing but dimerization, and that interferes in the synthesis of DNA as well as RNA. These are the chemical mutagens. We can further categorize into alkylating agent, base analogous, base modifying agent, alkylating agent. These are the alkylating agent, especially hydroxylamine. So original base is going to modified to formation of hydroxyl amino cytosine adenine pairing here. So hydroxyl amine cytosine, it's a derivative or modified base of cytosine, and that pairs with the adenine instead of guanine. So there is a formation of what? Transition from C to insertion of what? C to T here. So guanine is modified by methyl methane sulfonate, that is what? An alkylating agent. So formation of what? O6-methylguanine pairs with thymine here. We can see here the pair. This is usually normal pair with cytosine. It's a guanine. But after modification, it's O6-methylguanine. Sometimes pair with thymine also here. Base analogous refers to chemical compound are highly similar to DNA bases. These chemicals sometimes incorporate in DNA in place of normal base during replication. And these mutations are adding wrong base pairing or forming wrong base pairing. The most commonly used base analogues are 5-bromouracil and 2-aminopurine here. So these are the important part. The presence of 5-bromouracil enhances its tautomeric shift, that is from keto to enol form. Keto form is usual and most stable form, while enol form is rare and less stable form, like amino and imino form. Base modifying agents are also important. They work by modifying chemical structure and properties of the base. They are the nitrous acid. They deaminates a particular base here. We can say guanine deaminates or modifies to xanthine, cytosine modifies to uracil, and adenine modifies to form hypoxanthine, and they are forming unusual or non-Watson-Crick base pairing, and there is also forming, it's a predicted transition kind of mutations. Intercalating agents are actually important. They are also responsible to cause of mutation. They are generally thin plate like hydrophobic molecule, especially acridine compounds that bind directly or intercalating into a DNA structure, resulting there may be an addition or deletion of one or few bases. These acridine also responsible to cause of frameshift mutation in the coding sequence gene. You can see here mutation just because of intercalating agent or intercalating mutations. The formation of what results are the frameshift mutation due to insertion of one base pair. Tautomerism, that is that form is important. It's a change pairing qualities of the base, and that's why here pairing of purine with purine and pyrimidine with pyridine, that is nothing but tautomerism. So tautomers are actually alternate forms of the base that are produced by rearrangement of the electrons and protons in the molecule. So these may be caused by certain chemicals, and here DNA polymerase adds these tautomers during replication in place of correct nucleotide. You can see here thymine, it's normal keto form, adenine it's a amino form. There is a pairing of pyrimidine to purine and purine to pyrimidine. Cytosine it's amino form and guanine it's keto form. But in case of formation of tautomerization, thymine enol form is going to pair with guanine, it's unusual base pairing, and cytosine it's imino pairs with adenine, it's an amino. So cytosine is what? Again unusual base pairing with adenine. So these are the mutations caused by tautomeric. So result and predicted mutation is a transition kind of mutation, change of pyrimidine to another pyrimidine and purine to another purine. So another part is gene suppression or genetic suppression, that is nothing but reversion. So here this kind of mutation occurs by second mutation at same other site, and that reverses the harmful effect of the primary mutation. That is nothing but suppressor mutation. Nonsense mutation. So these are the tests to know what are the mutation and what are the mutagenic potential of the compound with the use of M test. Toxicity testing we can also use here. Role of mutation here. Where does the variation come from the first place? We can also know this part here. So these are the point mutation, revision, insertion, deletion, gene duplication. These are the phenotypic effect of point mutation. We can see here very clearly. Thank you all.